There are around 37 trillion cells in your body. They are growing, dividing, changing, and working to make you who you are. But not all cells are the same. There are many different types of cells with different characteristics that carry out different functions. The basis for these differences comes from molecules inside of your cells called proteins. Proteins carry out a plethora of functions and are involved in numerous processes that control cell characteristics. One of the most important processes, however, is the cell cycle. The cell cycle is the process by which cells grow and divide to make more of themselves. Cell cycle speed is controlled by checkpoints. These are points during the cycle at which the cell can stop and wait for the conditions to be right and for the signal that tells the cell to continue dividing. Cell cycle checkpoints are controlled mostly by proteins, most notably the cyclin-CDK complexes. The inappropriate regulation of these checkpoints, and therefore of the cell cycle, is what results in cell growth and proliferation diseases, such as cancers, which result from the formation of a tumor due to uncontrolled cell proliferation. Recently, a new cell cycle checkpoint has been discovered. This checkpoint is regulated by the interaction between the tuberin and cyclin B1 proteins. Tuberin is a tumor suppressor that integrates nutrient and growth factor signals in order to stop cell cycle progression when the conditions are not favorable. Mutations leading to a loss of function of the tuberin protein result in numerous cell growth and proliferation diseases, including tuberous sclerosis and cancers like breast cancer, lung cancer, renal cell carcinoma, and medulloblastoma. The focus of my research is to characterize the intracellular timing and localization of the tuberin cyclin B1 complex. To do this, I'm constructing a bimolecular fluorescence complementation system, or a BIFC system. BIFC employs the use of fluorescence in the form of the yellow fluorescent protein, or the YFP. What I've done is take both halves of the YFP and add them onto different parts of the tuberin and cyclin B1 proteins, so that when tuberin and cyclin B1 form a complex in mammalian cells, this protein forms a full YFP capable of fluorescing yellow. By analyzing where and when in the cell yellow occurs, I've been able to determine the cellular timing and localization of the tuberin-cyclin B1 interaction. Knowing how these two proteins interact could shed some light on the structure of tuberin, a protein for which we currently do not have a 3D structure. Finally, knowing this information will provide a more complete picture of the pathology of numerous cancers, as well as tuberous sclerosis, and will aid in the development of novel treatments to treat these diseases.